Hello, and welcome to our symposium titled Lessons Learned in Lockdown, Impacts of the COVID-19 Pandemic on Birds. My name is Michael Schrimpf, and I will be co-chairing today's session along with Olivia Sanderfoot. We are both members of C19 Wild, an international working group of ecologists studying the impacts of COVID-19 lockdowns on birds and other wildlife. In today's session, we will focus on what the scientific community has learned so far about the impacts of lockdowns on birds and the implications these findings have for avian monitoring and conservation. To start things off, Olivia and I have a talk that will set the stage for the rest of the symposium.
Thanks for introducing our symposium, Michael. I'm so excited to be part of this session. I'm sure our audience is acutely aware of the profound impact of COVID-19 lockdowns on communities around the world. In early April 2020, at the height of the first wave of the COVID-19 pandemic, more than half of the global population was under some type of stay-at-home order. At that time, lockdowns drove a rapid decline in human mobility that in turn impacted light, noise, and air pollution in major metropolitan areas. However, local impacts varied widely depending on weather, geography, baseline emissions, and the extent to which human activity was restricted. Regardless, around the world, many people paused to ask a unifying question: how are lockdowns affecting birds and our ability to study them? The COVID-19 pandemic has presented a unique opportunity to study how human activity and anthropogenic pollution influence birds and our observations of them in urban settings, an opportunity that ornithologists around the world have leveraged to gain insight into bird populations and effective conservation strategies. In this session, Michael and I will present a broad overview of research to date and spend a few minutes sharing findings from our own research. Then we'll hear from 4 other researchers who have also considered this research topic from several different points of view. We will conclude with a 30-minute discussion with the audience, and we encourage folks to jump in with questions and ideas. We know that this symposium will only scratch the surface of an incredible body of ongoing research on this important topic, and we look forward to hearing from folks who have additional studies or ongoing work to share in the discussion. With that, let's dive in. Research to date on the effects of COVID-19 lockdowns on birds has utilized a wide variety of approaches. Some researchers have relied on data from pre-existing community science programs such as eBird, whereas others have launched new community science projects to collect data during lockdowns. Alternatively, some teams have used data from long-term monitoring programs in their analyses, while others have established dedicated monitoring efforts. In addition, research to date encompasses explorations of impacts on birds in several regions around the world and represents investigations of many different taxa, including songbirds, corvids, wading birds, seabirds, and raptors. Today's symposium will highlight research on songbirds and seabirds, but we will share links to papers covering other taxa in the chat. Michael and I independently considered the impact of reduced human activity during COVID-19 lockdowns on birds in North America. Our 2 approaches have some similarities, such as using community science data, but also some differences, including the modeling approach we took and the geographic scale we explored. Together, our studies illustrate the value of linking bird observations with data on human mobility to explore how lockdowns influenced the detection, distribution, and abundance of birds. And we hope that by briefly outlining our approaches, we can help set the stage for the rest of the symposium.
Thanks, Olivia. So first, I'm going to briefly describe the work that I've been doing with a large team of people organized through Dr. Nikki Koper's lab at the University of Manitoba. This manuscript has just been accepted for publication in Science Advances, so it should be available to read in its entirety very soon. Our basic approach involved using eBird data from across the US and Canada to compare spring bird counts from 3 years prior to the pandemic to counts during the pandemic. Because of some important changes to how eBird users went birding during 2020, which we'll hear about later in the session, we put extra care into selecting the data that we used, choosing to subsample checklists so that the distribution of sampling effort and geographic coverage was as even as possible between those 2 periods. Then, rather than simply compare whether the counts of a particular species in a particular county went up or down in 2020, we focused our modeling approach on the interaction between the pandemic and these 5 indices of human activity here in the upper right. Each of these indices provided a slightly different perspective on how the pandemic may have been felt by birds, some of them directly, others more indirectly. The basic idea here was that if a change in bird numbers was due to the decrease in human activity, then that change should be different depending on how much activity changed. What we might expect to see then, for example, is that if a species that normally avoided roads, let's say, before the pandemic, due to, we'll say, heavy traffic or noise, counts of that species would have a fairly steep relationship with distance to road. But during the pandemic, regardless of how abundant the species was in that particular county on average, that is, regardless of whether this pandemic line is above or below the pre-pandemic line, if the slope of this relationship has changed, that indicates an effect of the pandemic. In this example, a negative relationship became slightly less negative, indicating that this species increased in relative abundance near major roads, where the reduction in traffic during the pandemic would have had greater effects. It turns out that many species, in fact, most species we looked at, showed some type of interaction with at least one of these indices, and that overwhelmingly the majority of those effects led to increased counts of birds in areas where the reduction in human activity was stronger. Here are just a couple of examples, both cases where bird counts increased in places where the pandemic had greater effects. There was also a lot of diversity in the responses within and among all the major taxonomic groups we were able to look at, including everything from waterfowl to raptors to passerines. And finally, after a lot of careful consideration, we determined that most of these effects were not likely only due to changes in the ability of birders to detect birds in areas with lower human activity. And we go through the evidence and reasoning for why we came to that conclusion in the manuscript. So what this tells us then is that the reduced human activity during the initial pandemic lockdowns across North America had pretty far-reaching impacts on lots of different bird species, likely causing a lot of birds to use developed areas a lot more, or at least in different ways. Our study was pretty broad in scope, though, so although we were able to show that the pandemic-related effects impacted lots of species across very large areas, it wasn't really able to tease apart some of the nuanced effects. Olivia's approach was similar, but some of the differences allow her to look at the issue through a different lens.
Thanks so much, Michael. Fascinating stuff. So last spring, my field season was put on hold due to the COVID-19 pandemic. And like many other researchers who found themselves in this position, I pivoted. Much of my research is focused on the impacts of air pollution on birds, and I wanted to come up with a new approach to this line of inquiry that would allow our team to collect valuable data while also keeping our community safe. With the support of my advisor, Dr. Beth Gardner, I quickly launched a community science project to monitor birds in urban and suburban areas under lockdown in the Pacific Northwest with two goals in mind. First, I wanted to collect data to learn more about how human activity and urban air pollution influenced the detection and distribution of birds as lockdown restrictions eased. Second, I wanted to create an opportunity for people to spend time, intentional time, outdoors birding, an activity that has brought me such joy and solace, and one I thought others may benefit from engaging in during increasingly uncertain times. We invited volunteers to sign up to monitor birds in their neighborhoods beginning April 1st, 2020. We accepted volunteers on a rolling basis. Participants selected their own monitoring sites, and we encouraged volunteers to survey birds on their own property or at a local green space they could safely access without violating public health guidelines. We asked volunteers to conduct 10-minute stationary point counts at their chosen site at least once a week from April 1st through June 30th. Volunteers recorded all the birds they detected during a point count using both visual and auditory cues and submitted their observations from each survey as a complete checklist in eBird. The outpouring of support from our community surpassed my wildest expectations. Our final data set includes 6,640 eBird checklists from 376 unique observers at 429 monitoring sites in Washington and Oregon, which is an absolutely stunning effort, especially considering the circumstances of the pandemic. We also conducted a survey of our volunteers last summer to collect additional information about their monitoring sites, specifically the availability of bird feeders and bird baths. This map shows the locations of all the monitoring sites included in our analysis. We had information on supplementary resources for the monitoring sites shown in blue. We used this data to build single species, single season occupancy models and explored how various aspects of urbanization influenced the detection and distribution of 46 backyard birds. We found that human activity, as measured by the percent change in human mobility, affected the probability of detecting 76% of study species. Furthermore, 50% of study species were more likely to be detected as human activity increased, possibly due to greater use of green spaces as lockdown restrictions eased. Our volunteers primarily monitored birds in urban and suburban backyards. This aligns with Michael's work. Some birds use developed areas more when human activity is lower. We also found that air pollution, specifically the ambient concentration of fine particulate matter, influenced the detection of 22% of study species. This adds to emerging evidence that air pollution is an important and often overlooked component of the observation process. Finally, we found that land use was a more important predictor of site use than bird feeders or bird baths across study species. Both Michael's analysis and ours illustrate the complexity of bird distributions at fine spatial scales in urban areas and highlight opportunities for future research on the impacts of human activity on birds. Our projects demonstrate the power of community science to answer urgent research questions and offer examples of how we can leverage pre-existing community science infrastructure, such as the eBird platform, to rapidly develop and launch new projects, even in times of crisis. We also show that studies can be designed to investigate responses at different spatial scales. Finally, I want to note that I personally found interacting directly with our volunteers to be incredibly valuable. This not only allowed us to minimize errors in data collection and reduce species misidentifications, but also gave us the tremendous opportunity to engage beginner birders, reach a wider audience, and learn from our volunteers' firsthand observations. I'm excited to talk more about our work and answer questions from the audience during the discussion. Back to you, Michael.
Thanks, Olivia. So our studies are only two of many that have leveraged community science or existing monitoring programs to learn more about how COVID-19 lockdowns have impacted birds and birders, which is why we're so excited to hear from the other four researchers today about their own approaches, and then to have a good discussion in our extended Q&A, which will take place on Zoom directly following the last talk of this symposium. We believe that this ongoing research will spark important conversations about how human activity affects the detection, presence, behavior, and abundance of birds, and help us identify practical monitoring and conservation strategies for a post-pandemic world. Thank you for joining us today, and we look forward to chatting with you later.